CPP, or conditioned place preference, is a behavioral paradigm that is commonly used to study the reinforcing properties of drugs of abuse in rodents. In this paradigm, a rewarding substance is paired repeatedly with an environmental cue so that the animal associates the cue with the rewarding properties of the substance and in turn develops a preference for the cue. In this video, we will be explaining how to perform a CPP protocol for studying the effects of ethanol, or alcohol, in zebrafish. In this CPP, the cue that fish will be associating with ethanol intake will be certain tank chambers, either a tank with blue dots on the walls or a tank with a white background. The two chambers are connected by a central alley that is brown, and there are removable dividers between the chambers. When running the experiments, the entire apparatus should be in a box with opaque walls to avoid outside distractions. Each fish will have its own apparatus. Make sure all fish are the same gender and around the same size. Make sure you are performing the experiment in the same time frame every day. The temperature is kept constant, which is the same temperature as the system that the fish are kept in on normal days, and that fish are fed at the same time every day, about 2 hours before the experiment. You must refer to the self-administration protocol video for other necessary forms of control, such as white noise in the background. There are 4 groups in this experiment, and each group should have at least 5 fish in them. The 2 main groups are fish that are ethanol conditioned versus the control fish, which are just conditioned with water from the system. Each of those groups is divided again. Half of each group is conditioned to the dot side, and the other half is conditioned to the white side, thus having 4 groups. This experiment has 3 phases: preconditioning, conditioning, which can be anywhere from 1 to 3 days, but in this case it will be 3 days, and then post-conditioning. On day 1, the preconditioning day, all chambers are filled with system water. Fish are put in the central alley, and then both dividers are removed at the same time. It is crucial that both dividers are removed at the same time. Fish are given 30 minutes to roam throughout the chambers, and the time spent in each chamber, including the central one, is recorded with a video camera and later inputted into an Excel sheet in an organized fashion, keeping track of which fish is in each of the 4 groups. Days 2 through 4 are conditioning days, where half the mice from the ethanol group are exposed to 1.5% ethanol concentrations in the blue dot side for 30 minutes, and the other half in the white compartment for 30 minutes. The water group is split the same way. This is the first conditioning session of each of these conditioning days. Fish are then put in individual tanks for 20 minutes before the second conditioning session. Chambers should be cleaned before the second session, or more chambers can be built. For the second conditioning, fish are reverse conditioned in the opposite chambers. This is called an unbiased counterbalance study, which makes the study more credible. So the ethanol fish receive water in the opposite chamber they were conditioned with ethanol in, and the water fish receive water again, but in the opposite chambers that they were first conditioned in. All of these sessions are for 30 minutes. All sessions should be recorded even though fish are confined to specific chambers. On day 5, the post-conditioning day, which is to test the expression of ethanol CPP, fish are put in the central alley, and then both dividers are removed at the same time and given 30 minutes to swim throughout the chambers, just like day 1. And time spent in each chamber is recorded with a video camera. The times pre- and post-conditioning are then compared to measure the efficacy of the conditioning. You can also refer to the behavioral brain research paper by Mather for an example of a zebrafish ethanol CPP.